Alrighties. Hi everyone. Happy Thursday. My name is Nat, and today we are taking a deep dive into neuroplasticity, looking at the memory. Now, heads up, this is a complex subject. This is not the easiest one we've done. So feel free to stop me, ask questions, and we'll do our best to get a good foundation knowledge on why is it so damn difficult to remember things. Now, we are a neurodivergent organization, so we specialize in helping people with neurodevelopmental conditions like dyslexia, dyspraxia, autism, etc. And a lot of people think, oh, well, dyslexia is about reading, autism is about communication. But one thing which kind of connects all of them is memory. Memory is sometimes some of the biggest challenges that you have through being neurodivergent. Hence why we're doing today. The more we know about why our brain works the way it works, the more we're able to support it. But this knowledge will be beneficial no matter who you are or how your memory is affecting you. I'm currently doing my master's in neuroscience, and I'm a neurodiversity specialist and support people with consultancy. And I'm also neurodivergent and also have a terrible memory. So it's an area I'm very interested and fascinated about learning more about. So as I mentioned, typically we talk about neurodevelopmental conditions, and while we are going to be focusing on that today, this is quite broad and will benefit most people who are interested in memory. Now, this is our lovely team, 80% neurodivergent, experts by experience. So we're an organization created by ND individuals for ND individuals and to help with consultancy. But we also deliver these free webinars every single week in order to, fingers crossed, benefit the community and spread knowledge far and wide and take really hard, complex subjects and make them digestible for everyone. Now, we record webinars every single week, and the one we did last week, which is really interesting, is the Science of Schizophrenia. So I do encourage you to check it out if you're interested in this area. Schizophrenia is a really interesting condition which kind of can affect your perception of reality. And we look in the neuroscience of it as well. So just go on our YouTube channel, type in exceptional individuals, or kind of like and subscribe, and I'll share a bit more about that later on. But now to why we've all come here. Formation of memories in the brain. Now, our brain is continuously remembering memories. Like, we think we remember exactly what happens, but more often than not, we remember the memory of remembering. So over time, the memory gets faint or distorted. And ultimately, what is memory? Just a bunch of chemicals, electrical signals. Also, the exact same things which often result in certain neurodevelopmental conditions like early on. So there's a strong overlap and link between the two. Oh, Vinod said I had a head injury in 2008 and a major ischemic, probably pronounced that wrong, stroke in 2012. But I was saved in Charing Cross Hospital, and I've had hardly any physical damage, just very serious cognitive effects. Well, thank you for sharing that. I appreciate it. And this is some of the things as well. Like, memory formation can be something which is a challenge through developing early in life, or it could be something which is acquired through, say, a particular injury. So the ways it affects people, it does translate for both. Now, what I'm hopefully going to cover today, and again, these aren't the easiest subjects, so hold on tight, is synaptic plasticity and long-term potentiation. Now, if you've never heard of that, I am going to explain it, so please do not worry. Neurological basis of memory, so where does memory originate in the brain? And properties of long-term potentiation, what a mouthful, and how it kind of relates to learning, because obviously learning and memory, two peas in a pod. Now, to start off with, let's get right back to the very beginning of neuroscience. Good old neuron. A neuron is literally just your nerve cells in your brain. Now, we have thousands and millions and maybe even trillions, but definitely millions of brain cells in our head. And these are little thingies which send messages from one to the other. Now, on here, you can see how it's sending little bursts of electrical signals up and then it sends chemicals out the end. I am not going to tell you what each bit is because I want to see if you know any of them before. So on this image of a neuron, if you're able to, I want you to click on the one which you believe is a synapse. And if you've not been able to join, just say in the chat, is it 1, 2, 3, or 4? So which image is showing a synapse? If you have no idea, just click one and we'll see how it goes. Okay, interesting. We've got one in already. Now, that isn't actually the right one. Oh, okay, the last one. Yes, it is the last one. So the first one is our dendrites, and essentially that's like the arms, and they are what kind of catch other signals and brought in. So lots of dendrites, meaning getting more information. Then you've got the middle one, which is the nuclei. That's like the brain of the brain. Important thing. This is where like our DNA is kind of embedded. Then we've got the axon in the middle. This is like the long cable which takes the information, and you'll see we've got these squidgy bits on it. That is called myelin sheath, and that's kind of like the insulation which allows the electrical signals to go really fast. And at the very, very end, we've got the synapses. Now, the synapses is actually that tiny bit of gap. So when one neuron connects to the other neuron, they're not actually touching. There's a tiny, tiny little gap. And that is going to be really important because the way it works is when our body gets like so much kind of electrical charge in one, there's like this tipping point, and then it sends a rush of like an electrical signal through the axon. And then when it gets to the synapse, there's that gap. And then it's chemicals that take over. This is going to be really important because it is through this bit, the synapse, where people believe that memory is formed and when challenges can arise. And well, we are just so you know, I don't send the slides unless people personally request them, but we do record all these sessions, so you will be able to catch up. Otherwise, what good is a memory session if I don't give another way for people to remember? So yeah, we'll have this recorded. So let's have a bit more of a look into what a synapse actually is. And as I mentioned, it is a small gap between two neurons that allow them to communicate, sending electricity. So you've got to remember, electricity, then chemicals, then electricity. And it's this tiny little gap. Now this is going to be a bit of a pain for us because essentially little chemicals are already kind of stored at the end where the synapse is, and they get kind of pumped out. Different chemicals are like different messages, and then they kind of get absorbed or sucked up on the other end of the dendrites. So essentially, our brain is continuously communicating with others. And remember, we have thousands and millions of these, but they are very, very important. So plasticity is the brain's ability to adapt and change due to experiences and injuries. Now, plasticity is something we're going to be talking about again and again and again. We talked about how the brain has different connection signals, one after the other, after the other. Now, they can change. They can grow an extra arm, an extra leg, and they can connect and join. If one gets damaged or cut or die, it can remove. So the brain is continuously developing and moving around. Obviously, when we're younger, the brain is developing a lot, and the brain is really placid. If you've ever heard people say, oh, children can learn things so much faster, or I wish I learned a language as a child, I would have picked it up a lot easier. There's real science behind that, because the brain is continuously, think of it like a little tree branching out new shoots. A young tree will be growing more branches, and if a branch gets cut off, it's able to regrow a new branch a lot easier. But an old tree that's been around for a long time will not grow new branches, not nearly as frequent. That's essentially how our neurons are. So we've got birth, then after 1 year, 3 years, 5 years, the brain is continuously rerouting and redeveloping. Another way you can look at this is that these kind of connections, they're very much like muscles that we have in our body. The more you use a muscle, the stronger it gets. The less you use that muscle, the weaker it gets. And when you're younger, you're exploring the world. Everything's new and exciting. So your brain is continuously using those muscles. But when we get older, if we don't practice like brain training and look after ourselves and stay active, it can get kind of weak. And the brain's ability to form new memories diminishes. And essentially, that's all memories are. It's the plasticity. It's when we see a new experience, our brain is creating a new branch and a new memory, a new branch. And when we want to remember something, we're kind of retracking those branches. But if those branches are a bit severed or kind of just not functioning properly, we can't get access to that memory. Hopefully that makes sense. But please do let me know in the chat and the comments if you require a bit more information. I'm happy to go into as more detail or as little information as you all think is needed. So to go in a bit deeper, we are going to be looking at neurons. So these little images here, these are the neurons. We're used to them. We saw them before. We saw the little dendrites, the little spiky bits, and we saw the synapses at the bottom with the chemicals coming out at the end. Now, synaptic plasticity, remember, is just about the bottom part of a neuron, and plasticity meaning kind of being flexible, being able to mold, being able to change and adapt. So sorry, I'm very dyslexic. I'm like the worst science teacher ever, but away we go. Synaptic plasticity is changing neuron connections for better or worse communication. And that is a really key thing. You might think that when the brain is kind of changing its arms and legs around, it's always going to be better. Not necessarily. Sometimes it might make it worse. A good kind of visual image I also see is if any of you watched Harry Potter, when you've got those stairs that are always moving around and the stairs are never stopping in one spot. It's kind of like that. You never really know where they're going to go. So in a healthy brain, you can see all these neurons are looking beautiful, one connecting to the other, all lovely. Now, let's say you get a bit of brain damage. Oh, you've lost a brain cell. A neuron has been died or damaged. Now, obviously, neuron A can no longer communicate with neuron B. That's going to be an issue because we really need them to communicate. But because it's damaged, it will just kind of reroute itself, just like your sat nav when you take a wrong direction and go directly to C or D. And then when your brain is recovering, because you can recover, it will then get stronger. So in this second image, if you see this little arm here, see how weak and skinny it is. After a while, when it has been using it more and more, you can see in this last one, it's a lot thicker and stronger. We often see this in like life, let's say you become blind, you become a lot more better with your other senses. Or if you have any sort of impairment, your other senses kind of step up and you normally become a bit more attuned or a bit more aware. Your brain, neurons, are doing the exact same thing. They're very resilient and they're very resourceful. While the brain can grow new neurons, it's not well understood, and that's called neurogenesis, but it's still a very emerging field, and it's not like we grow a lot. So essentially, the neurons you have when you're born with, you don't really get many more, and that's a worth thing knowing. So while the brain can reroute itself, you're then putting extra stress or more energy resources in one, which would have been distributed between more of them. So the brain's an amazing thing. And I know some of you mentioned about having, say, a physical or an accident which kind of resulted into poor memory. You're probably in this middle stage now, and you absolutely can get to the second stage. Now, for some people, that happens naturally. For other people, they have to put more kind of effort or work in. And for others, it's completely out of their control. Going even deeper, and if you see these images and you're scared by them, please don't be. I will try and break them down as much as I can. So on here, we've got synaptic plasticity is how the brain adjusts the volume of messages between cells and aiding learning. So like I used the analogy of the muscle, another one you could use is the volume. So if you want the message to come around loud and clear, you might dial it up. If you want it to be less thing, you might dial it down. And that's a really useful thing to have. So as here, we've got our neuron, and this is a synapse. And as you can see, glutamate, which is a chemical, and it can kind of come out and be absorbed. But we can either send them out at a faster rate, a slower rate, and it's so kind of dependent upon what we kind of want to get across. So just remember, when we want to send signals to another part of the brain, this can be more or less. And there are challenges in memory for both of them. If you have too many signals, challenges. If you have too little, challenges. And if any of you did attend last week when we talked about schizophrenia, you'll also notice that it's very similar in terms of the deficits, either having, say, too much dopamine, too much glutamate, depending on what chemical or neurotransmitter, which is the name we're going to be using, it can have very different effects. Now, in terms of how these chemicals are sent across, they can normally be split into 4 different names. So we've got long-term potentiation, LTP, and do not worry, I am going to talk more about these. We also have long-term depression, LTP. I'm guessing you can imagine this means when you have less. The first one's more. Then short-term potentiation and short-term depression. All it means is one is when you get too many chemicals, and one is when you get too little chemicals. One is when it's a quick one-off. The other is when it happens over a longer period of time. So depending on what one of these you have, it will depict whether or not you have a mental health condition, memory challenges, communication problems. They can really result into many, many different ways. We honestly do not know everything yet, but we are actively learning. So on this brain, I want you to put a little pin, if you're able to, on where you believe the hippocampus is. Why? Because the hippocampus is believed to be absolutely integral to when it comes to forming new memories. So the synaptic plasticity is mostly studied in the hippocampus. So in terms of where does the brain make new connections, that is normally done in the hippocampus. I like the word hippocampus. It always reminds me of like a college campus filled with hippos, but it is not like that at all. So I can see we've got a few dots all over the place. The correct area is this part, the green part of the brain. And this is the hippocampus. And this is where we are going to be putting all of our focus in today, because this is the area of the brain which is predominantly in charge of memory. Now, why I say predominantly is that there isn't one part of the brain which is solely focused on anything. Like the brain is very flexible, like we've already discussed. But if things are all going to plan, there are areas which we see more activation, particularly when we do scans like MRI. So here we are. Here's a hippocampus. It looks like the little Nike tick symbol. And this is the brain structure in the temporal lobe. And apparently it was named after the Greek word for seahorse because it looks a little bit like a seahorse. I don't really see it myself, but maybe if you squint and kind of turn to the left, maybe you can see a seahorse. However, it's a useful way to try and remember it if you ever see this image of a brain again. Now, a really interesting case example, which got people actually thinking about the brain and memories, was the case of HM. Have any of you ever heard of this fella, HM? Oh, Vinod, this is a really interesting case example, so it's always worth researching more about this one. So his name was Henry Gustav Morlason, but everyone just called him HM because people didn't talk about people's names until after their death. Now, the interesting thing about Henry is that he was a patient who underwent brain surgery, and that surgery resulted in severe amnesia. So amnesia is when you forget. So why did this happen? Why did brain surgery make him forget things? If we can work out why he forgot, maybe we can work out how we can support people to remember. Oh, I can see some of you had heard of him. Now, he had epilepsy, a condition causing repeat seizures like brief electrical storms in the brain. So remember we talked about the neurons and how they send electrical signals in order to communicate? Epilepsy is when it sends those signals at a way too much rapid rate. It's like too many signals. Imagine if you kind of get someone's phoning you and then like 10 other people are phoning you at the same time. Your phone wouldn't know what to do. It would just kind of give up. And that is kind of what happens in epilepsy. So here you can see the normal neuron, how the electricity goes at a kind of very standard rate. But with an epileptic neuron, you'll see how it goes way too quick and also not as frequent. So this is essentially what happens in a seizure. And again, a good way to picture this is like a little electrical storm in the brain. The brain is all peace and calmful, and then seemingly out of nowhere, electric storm. And this is what happened to Henry. So HM suffered from severe epilepsy. Surgeons tried to remove the hippocampus because you've got two different hemispheres in the brain, and one was sending signals to the other one way too rapidly, and it was kind of short wiring the brain. This wasn't any good. So they thought, well, the hippocampus seems to be the problem area. Let's just remove it and see what happens. Maybe it'll fix it. It worked for the epilepsy. Thankfully, because they removed that part of the brain where there was a lot of neurons that were sending signals back and forward at a rapid rate, by removing it, it did sort out the epilepsy. Great. However, it had an unintended consequence, and that consequence was impaired memory. He just wasn't able to remember anything anymore. And it was through this study, which was, sucked for Henry, but for the rest of us, we were able to learn that the hippocampus is vital for learning and memory. If we get rid of it, the brain is not able to remember, and you have amnesia. This image you can see of the normal brain, and you can see the kind of light grey patches, the hippocampus, and in Henry's brain, they have been removed. And while he was still able to live, it didn't affect him in other ways. It did affect him in memory. So it's clear that, is this the way we want to be solving epilepsy? Probably not. It's like solve one problem, you gain another problem. Now, the way we study the hippocampus today, we try not to cut the brain up too much. It's not great for business. What we do is use little electrodes, and these are like little metal spikes you kind of put in the brain. I've never had them done. I don't know if any of you have. They don't look particularly great, but essentially they're a way of looking at the brain, and they can send little jolts of electricity which can kind of control the brain. Think of it like installing an off-on switch. It's they use them a lot in mice, but they do use them in humans. The downside is they are invasive, so they're obviously not people's preferred method if they can help it. So it's a little device that detects or stimulates brain's electrical activities. So detects meaning it can see how rapid the brain is functioning. So if you have epilepsy, it's able to record that, but it also can control it as well, like a little floodgate again, turning it on or off. So now we know how we're actually doing it. This is how we ended up with this data. So when they use these signals, they've used them in mice studies, they have also used them in human studies. The electrodes will stimulate synaptic currents. So remember, synaptic currents is the electricity going through the neuron. And what we can do is kind of force one to happen. Like say, go on, we want to see what is actually happening in the brain. So we send a little jolt of electricity in order to stimulate the brain cell firing. Now on here, you'll see different images, and I want you to take a guess on which one you think is long-term potentiation. This is when the brain is firing signals at a rapid pace for a long period of time. And this is one of the key things which helps create memories, but also can make forming memories more difficult. Okay, interesting. Now, if you have no idea, honestly, do not worry, I wouldn't be surprised. But when I show you what it is, it makes a lot of sense. So it is, in fact, the middle one. And you see these little dots at the bottom. That is when the brain is acting very kind of conventional. Actually, no, let's start with the first one. So the first one, the brain cells are working how they should. Now, when a signal is being sent to the brain, you'll see a massive spike, and that's essentially electricity. And that's a little spark. And then the brain is active, it has sent the message onto the next neuron. And then you'll see a slow decline back until it gets to that normal level. So the brain is working nice. Oh, signal, spike, then it goes back to normal, business as usual. That is how the brain is supposed to work. But when we have long-term potentiation, this is when the brain is working normal, spike of kind of electricity, and then it goes down, but it doesn't go completely back to normal. It stays still pretty active. And that, they believe, is the kind of sweet spot of when we learn memories. Now, the last one is, remember how we talked about long-term depression? That is when we have a decrease. So the brain is working normal, and then there's a decrease in terms of the signals and frequency that the brain is receiving. Those two different things will result in different characteristics or different deficits or different ways of forming memory. Today, we're focusing on the middle one, because this is the one that things can either go really well in or really not well. Okay, I hope you're all still following with me well. I appreciate this is a bit challenging, some of it. So long-term potentiation is when neurons keep chatting to each other a lot. It's when neurons become like best friends forever, and they help them to talk more easily and quicker. So when people get very used to it, they can keep chatting. So here is a normal neuron. As you can see, the neurotransmitters, the chemicals, are kind of going out, and then they'll be absorbed by the dendrite. But with long-term potentiation, they've got like a more rapid amount, and they stay around for a lot longer. So the longer that these neurotransmitters hang around, the longer they can be absorbed but from the other end, and the more likely the signal is actually going to be retained. Think of it like this. If I held a picture up of a duck, you might remember the duck. If I remove the picture after a second, you might forget what color that duck was. But if I held that picture of that duck up for like 2 minutes, the chances of you remembering that duck is a lot longer, because you have been visually exposed to it for a longer period of time. Very similar to this. The longer that these neurotransmitters are hanging around, the much longer they can be absorbed and retained. So here is a synaptic model of the memory in the hippocampus. Now, the way we study the brain often is in animal studies, such as mice, mainly because we don't really like using them on ourselves. Poor mice, but they are a really useful way in terms of having a human-like model that we can kind of test on. So long-term potentiation was thought to be a mechanism of storing information in the brain. And now this is interesting. What we say for, because it's a developing area. But essentially, the longer the potentiation, the longer we're able to store the brain in. It has like a better write speed. If you try to write things to the brain too quickly, it doesn't get a very strong image. If you spend longer writing that message to the brain, it's far more likely to stay in there. Now, a really classic example of this is through Pavlov's conditioning. Now, maybe some of you have heard of Pavlov, really kind of a influential guy in the field. He was learning about conditioning and how we can get people to do what we want. It's a really classic study. So in here, you've got this dog and there's a food. Now, we know when dogs see food, they salivate, they start to dribble. They're like, oh, I love that food. So what Pavlov did is he said, I'm going to ring a bell. And when I ring the bell, dog does nothing. But if I ring the bell, every time I feed the dog, he'll start salivating. And when I ring the bell afterwards, even when there's no food around, the dog is still going to salivate and dribble. Because the dog has remembered that every time he hears the bell, food is coming. And this is how he was able to train the dog. And in a neuroscience perspective, that dog's connections in its brains are getting stronger. And every time it hears a bell, it is making that strong connection in order to think, it relates to food. That is how memory works. And that is basically what long-term potentiation is. But every time we do something and there is a signal, the brain is getting rewired in terms of that plasticity, and it forms stronger, more robust connections. Long-term potentiation. We need to go into even more detail on this. I know it's probably a term that's quite new to a lot of you, but this really is the key area that memory evolves around. Now, in here, have a guess. Do you know which one of these little chemical signals is demonstrating glutamate? Now, glutamate is a neurotransmitter. Neuro as in brain, transmitter as something that sends a signal. So when I talked about a neuron sending electrical signal and then sending a chemical signal via the synapse, those little chemicals are called neurotransmitters. And I think there's like 55 of them. There's a lot. There's a lot of different potential chemicals, and different ones can cause different effects in the body. It is, in fact, the last one. But honestly, I don't expect any of you to remember that. The first one, we've got like dopamine. Glutamate is a neurotransmitter that helps brain cells communicate, playing key roles in memory, learning, and quick reactions. So in this one, glutamate is the chemical that we are most concerned and interested about. It's important to know that while it is predominantly in memory and learning, it's not the whole story. There's more to know. Now, this image kind of shows you what's going on. You've got one neuron and the next neuron. And as we mentioned previously, there's a gap. They're not quite touching. And they are sending the little messages, think of those like little envelopes back and forth. Well, not back and forth, just one way. And sometimes it gets resucked back up. Now, each of these little messages are going to be different chemicals. And when glutamate is sent, that is essentially telling the brain, hey, we should probably remember this. To break it down further, glutamatergic, so many long words, synaptic transmission is this. So glutamate used in synaptic transmission. So glutamate is a messenger chemical. Then it binds to receptors on postsynaptic neurons. What does that mean? Essentially, the chemicals come out into the synapse and they join to the receptors. These are these kind of little things. They kind of like hug the chemicals and absorbs it. Postsynaptic just means post as an end, synaptic, so it's the other end. This is presynaptic, the other side is postsynaptic. So it's going across one end to the other. Nice and simple. Then it triggers an electrical signal, passes to the other neuron. So when these chemicals have been taken here, you get a big build-up, and it's like, all right, we've got too many chemicals now, and it releases the chemical. Like, you know, when you've got racing dogs and they lift the barrier up and the dogs go sprinting. When there's enough dogs, then they can go. If there's not enough, it's not going to trigger that reaction. Changes in receptor shape for improved communication. So these little receptors, they also play a big part in memory. If they are the right shape, essentially, yep, the gate can open nicely. But if the gate isn't really open, if the gate is broken, if the gate's a bit dodgy, then you're going to get less efficient messenger signals. So not only are the chemicals important for neurotransmission and in terms of memory, not only is the synapse, the neuron, important for memory, but we've also got the receptors, the things that kind of catch the signals and then sends it through the body. So you can see why working out more about memory is challenging, because it's not like we really know. It can be many, many different things. And as a doctor, they aren't able to know. Most often it's a process of elimination. They're very good at knowing what isn't the problem and not very good at knowing what is the problem. Now, you've been a quiet bunch, so I assume this is making sense, but do let me know if you want more information. So quick question to all of you. Do you know the difference between the synaptic protein and a neurotransmitter? If not, don't worry. They are an interesting thing. A lot of people get confused between the both, but they play different roles. Okay, 100% no. This is good. It means we can kind of teach it. So in our little neuron, in the synapse, you can see that we've got different kind of like chemicals around. Now, the neurotransmitter, as we know, is the chemical, whereas the protein, think of that kind of like the scaffolding, like the structure that kind of gives the synapse its shape. So synaptic proteins have two main functions. They regulate of neurotransmitter. They're kind of like the security guards. They're like, okay, yeah, let's see your ID. All right, glutamate, off you go. Dopamine, cool, off you go. They kind of do the regulation. They help to create and maintain the physical structure. Again, very much like scaffolding. They are needed in order to give the structure rigidity. They enable neurocommunication by organizing and maintaining proper synapse. So without your proteins, your neurotransmitters and your synapses aren't going to be very strong. They are the maintenance crew. They are the janitors. They are the people who keep everything running smoothly. Where neurotransmitters, on the other hand, these are the chemical signals that carry the information. So while a lot of time they can get confused, very important. And now we've got another thing to add to the equation with things that could possibly go wrong when it comes to forming new memories. Not only do we've got to keep tabs on the chemicals and the synapse and the neuron in total, but also the proteins, the janitors of the synapse. So as you can see, first of all, you think, okay, it's just my brain. Oh, now it's the brain cells, the nerve cells, the neurons, the synapses, the neurotransmitters, the proteins. It gets smaller and smaller and smaller. And honestly, there's probably more that we haven't even realized and discovered yet. Now, in terms of these like proteins, we have enzymes. And do you know what exactly an enzyme is? It's probably a word you've heard of before, but have you actually thought what an enzyme is? Now, the options are as followed. A type of fat that provides energy for the body, a structure within a cell that stores DNA, or a type of protein that speeds up the chemical reactions in the body. Nice, I'm glad all you did that. I think I've kind of gave it away anyway. So it is, in fact, the last one. So when we talk about an enzyme, it's just a type of protein. Remember, the protein is like the scaffolding or the security guards in the synapse, and they are absolutely essential. Now, there are lots of different types of protein. It's not just one, there's lots of types. And one type of it is an enzyme. And they essentially regulate how fast things are moving, helps with reactions. So you've got your enzyme here, and think of the brain as often like a jigsaw puzzle. If something fits, it works. If it doesn't fit, it doesn't work. So now that we know what an enzyme is, we can push a little bit further. Now, are you ready to continue? We've talked a lot of subjects and probably a lot of new terms. So we know what an enzyme is. How does these kind of scaffolding proteins in our brain, how do they actually relate to long-term potentiation and memory? All right, so we've got, oh yes, one of you is as ready as you can be, and another one is ready. That's good enough for me. No one's run away in here just yet. That's always reassuring. All right, we have an enzyme, and this I'm going to tell you anyway, but genuinely, I don't think you need to remember this, but it's interesting. We have an enzyme called CAMKII, and this contributes to long-term potentiation production, important for learning and memory. So this is our dendrite, this is a synapse, and this is the pre-synapse. And as you can see here, there's a lot going on inside our neuron. Now, if we have the enzyme called CAMKII, the likelihood of us having a more long-term transmission increases. So this is good. We want this. But believe it or not, this is where you might have less of them as you get older, if you have brain damage, neurodevelopmental conditions. Now, in terms of long-term potentiation, it can be divided into 3 phases. It's not all equal. First of all, you got the introduction. Remember, this is when the brain is kind of going all normal, and then it gets the jolt electricity, and then, all right, we've just had a sudden burst. That's the introduction. Then we have the early stages. This is when you start to see a slow decline, and the late stages is right at the end when it's finally running out of steam. So different stages can also equal different potential areas which might be involved in terms of memory formation. So we are getting really, really far through the looking glass now. Back to long-term potentiation maintenance. Though it is an important part of how the brain stores memory, it's not something which does it on its own. It needs continuous maintenance. It needs lots of repairs. It needs to be kept ship and shape. And this is where the proteins and the enzymes really come into their own. Long-term potentiation, remember, LTP, maintains memories. Memories aren't something which you create and just stay stagnant. They are something which our brain must continuously work on. If it isn't maintaining the memories, we lose the memories. And it's kind of an interesting way of looking at it. It's not like when you take a picture and the picture's there forever. It's like, imagine you take a picture and unless you're kind of cleaning it, scrubbing it, maintaining it, it will disappear forever. And maybe like kind of paintings where we have to look after them. Now, if you have strong synapse connections, remember like strong muscles in the brain, then it's far more easier to maintain and look after these memories. We can form and store long-lasting memories. So people who might have weak connections might struggle to form longer memories or shorter memories. And enhances and maintenance can increase these neural pathways. It really is very much like building muscle. Use it or lose it. And unfortunately, some of us do not have a choice over that matter. So this image, again, don't worry too much about it, but it just shows you there's a lot of things that's happening inside our brain cells in order for us to remember. It's not an automatic process. There's a lot going on under the hood. Now, the question is long-term potentiation needed for learning and memory? Now, from what I've said today, you probably think, absolutely, but a lot of these things we're still learning about. It definitely is, but to what extent and how it can be manipulated, treated, or supported is still an emerging area. There's some studies that we're doing in order to really get down to this and find out what's going on is by doing memory tasks in the old hippocampus. Remember that kind of little seahorse in the brain. First of all, by removing of the hippocampus, we know that it impairs memory in epileptic patients. That was done ages ago with HM. Okay, we know that. Animal experiments are needed to understand hippocampus removal effect. So we're still doing it now. We're just no longer removing it in humans. So maybe we're getting a little bit more ethical, but still very useful. And lastly, we're focusing on mice and studies of the long-term potentiation and memory. So studies are still very much underway, but we have moved over to animals. Now, how can we actually use mice? Well, I'll show you in a moment. But in terms of this, it's understanding a bit more of memory. Out of long-term memory, solid-term memory, short-term memory, or working memory, do you know which one isn't a type of memory? Yes, solid term. So there's different types of memory. And remember how we talked about how so many different things are going or happening in our brain in order to form memory. That really depends on whether we have short-term memory, long-term memory, working memory. So this study here is a really interesting one. And this is just an example of how we study mice in order to improve our understanding of memory. So you've got this little mouse here, right? Imagine this big pool of water. We've dumped the mouse in the water. The mouse is swimming around, but the mouse isn't able to get out of the water. It's like, there's no ladders. This is awful. And eventually, in the water, there's this like pillar. And the mouse can't see it because the water is this kind of like milky color. But when it gets onto this kind of like pillar, it's able to stand up and survive. Whew, all right, this is brilliant. And what they do is they then take the mouse out, and then they put the mouse back in the water. Now, if the mouse is able to swim straight away and get to that little platform, you know that that mouse has remembered correctly. But if we've done little like experiments, we try to like change how the brain is operating, the mouse may not remember. So this is how we know whether or not an animal has actually remembered. Is it a bit cruel? Yeah, but it's kind of a necessary evil at the moment, but really interesting how they kind of judge. Obviously, we know that animal models aren't a one-for-one with human models, but in terms of consistency, mice are a very good case example for that. Now, I'm going to summarize now because we've nearly come to the end, and I appreciate this is a really challenging subject, so well done for sticking at it this long. The key takeaways is knowing that when it comes to memory, we've got to look at the synaptic plasticity. This is the brain's ability to form new, stronger connections. but it can also make weaker connections. It can go either way. So synaptic plasticity is the brain making new connections. Then we need to have a look at synaptic transmission. This allows the neurons to communicate through electrical to chemical signals across synapses. So one sending a signal to the next neuron, that is another key area we must always look at when forming new memories. And if we're looking about where in the brain, we're typically looking at the hippocampus, that little seahorse. That is where the majority of our learning, memory, and plasticity happens. And last of all, in terms of how it's happening, it's the long-term potentiation. It's when the brain is sending a lot of signals, a massive spike, and rather than decreasing straight away back to normal levels, it's having a slow decline. And that slow decline is when the brain is storing the memories and creating those strong connections. So these 4 areas are something which I encourage you to explore more on to learn about how the brain is focusing. How you make this relate to your own personal life, I'm interested to know. Now, as we come to the end, I want you to know if any of you are in the workplace currently and do have memory challenges, you are most likely eligible to get free support from the government for a scheme called Access to Work. And essentially, this is where you can get free equipment and training to support you in the workplace. If this does sound like you, honestly, get in touch with us because it's what we do on a day-to-day, so we might be able to help you. Now, any last questions from today? Anything that you learned or want to share or thought? Now, you can either answer now, or I can give you my email address at the end, and you're welcome to email me afterwards, and we'll kind of go from there. Oh, we got, are there any other parts of the brain linked to memory? Yes. Now, again, in terms of the brain, it's a very plastic, malleable area. So when they do brain scans, they'll kind of look where certain areas of the brains are lighting up. Now, the hippocampus has the most activity, but it doesn't mean there isn't activity anywhere else. It's like saying, what part of the brain is purely responsible for vision, communication. Though we can pinpoint an area where there's the most activity, you do see a bit of activity all over the place. So it's not like an exact another area, but let's say if you were to remove another part of the brain, you would still notice an impact on, say, memory. It just might not be as substantial. Really good question. Oh, you're welcome. Now, if you found today useful, we did record it and it will be uploaded to our YouTube channel. So do check it out on Exceptional Individuals, just Google it. Now, we are doing really interesting subjects on neuroscience every single week, and we've got some really interesting ones coming up. And it will also give you a chance to re-watch and to retrain those neuro pathways. Remember, the more you watch, the more you engage, the stronger those pathways get. It's why it's really great to comment, share, interact, because you're just building stronger pathways. Our next webinar for next week, if any of you would like to come again, we're looking at autism, cognition, function, and ASD. So we've talked a lot about memory today, but how does this affect autism? Why do people with autism struggle with cognitive functions, kind of like just navigating the world around them? We're going to be looking, because a good example is this image here. What is it an image of? Is it an image of a woman, or is it an image of a saxophone, is it a saxophone? A saxophone player. And depending on which one you saw first says a lot about the way your brain is wired. So this is a really good continuation. Here are our contact details if you'd like to learn a bit more. My name's Nat, N-A-T, and my email address is nat@exceptionalindividuals.com should you have any follow-up questions. I really hope you found this useful and got a couple little gems out of it. And hopefully I'll get to see you all next week. All right, everyone, we're done. That was a lot. That was a very hard subject. So thank you so much for staying with me. Oh, thanks. It's been very useful and you've been a great teacher. Thanks so much. No, I really appreciate you attending. And I hope the rest of you found this useful and able to benefit your life in some shape or form. All right, everyone, I'm going to head off now, but I hope you have a great rest of your Thursday. And yeah, see you next week. Thanks, April, again for recorded. Oh, thanks, Fleur. Appreciate it. Thanks, Vinod. Thanks, Sophie, Charlie. Thanks, everyone.